Hi, I'm Jessica Lynch from UCLA. Thank you so much for inviting me to speak on International Humboldt Day. Humboldt was an amazing explorer and polymath, and he provided substantial contributions to our understanding of primate taxonomy and diversity in the Neotropics. He described Capuchin monkeys, and in fact this squirrel monkey up at the top middle, Cimeri cassequariensis, is named Humboldt squirrel monkey in his honor. I also wanted to thank Dr. Alberto Gomez Gutierrez, who sent me some of his scholarship on Humboldt, including information on primates Humboldt described on the Orinoco and Magdalena rivers in his work in 1811. In 1812, Humboldt published more species accounts, and in the book Primate Taxonomy, Colin Groves also credits Humboldt with the discoveries of species of Mico, Calothrix, Seguinus, and Sapajus. In fact, Humboldt describes species in almost all neotropical primate genera that are recognized today. I can't overemphasize the debt we owe to individual explorers, scientists, and observers for our knowledge of primate diversity, and I will try to highlight other key researchers in the field in my talk. I myself work as an evolutionary primatologist, studying how the astonishing diversity we see across neotropical primates has evolved. I'm interested in how these differences in morphology and behavior have accumulated over time and expanded across space from a single common ancestor. Today I will give a short overview of some questions in biogeography for the Platyrrhini, or neotropical primates as a whole, then focus on my own work with one family, the Cebidae. The Cebidae includes the squirrel monkey, genus Cimeri, and the capuchin monkeys, the robust capuchin Sapajus, as well as the gracile capuchins Cebus. Formerly, the capuchin monkeys were lumped together as one genus, Cebus, but now they've been split into two genera, and we'll talk a little bit about why. And all these three genera have wide and overlapping distributions in the Amazon, and all three are also found in other biomes. So just to give you an overview of my talk today, I will talk first about neotropical primate evolution and biogeography, then look specifically at squirrel monkeys, capuchin monkeys, and in the end, think about how phylogenetics and biogeography inform taxonomy and conservation priorities. Well, what are the Platyrrhines and how did they get to the Americas? Neotropical primates represent one of the most successful mammalian radiations in the neotropics, and all living Platyrrhines in Central and South America stem from a single common ancestor from about 22 to 23 million years ago. I wrote about the monkeying of the Americas in a review paper on primate biogeography in the Neotropics, and I would be really excited if any of you want to contact me. Here's my email address. I would love to send you a reprint, discuss this paper or my talk in more detail. So if we think back in time, we can see the changes over time in landmass geography and relationships to each other. And it turns out that South America was an island continent in isolation from other continents from about 60 million years ago. And I want you to note a few other things, that during the Cretaceous at 65 million years ago, it had already separated from Africa, but at that time Africa and South America were still a lot closer to each other than they are today. You can also see that the last land mass that South America was connected to before becoming an island continent was Antarctica and through to Australia, and that explains why marsupials were an important early radiation across both regions. So what was on the island continent of South America when it was separated? There were non-therian mammals like Thalacosmilus. There were many different kinds of marsupials. There were also xenarthrans, which were the ancestors to the sloths, anteaters, and armadillos that we see in South America today. And there were also various kinds of ungulates. And then you get the lucky arrivals. Neotropical primates, or New World monkeys, and caviomorph rodents, which include the capybara and the guinea pigs, their ancestors both arrived in South America from rafting over on current from Africa on large vegetation masses around 35 to 40 million years ago. These ancestral populations led to all the diversity today in these groups. Presently, there are 372 primate plus caviomorph species versus 136 marsupial plus xenarthran species in South America. So these lucky arrivals ended up with giant radiations and are now two of the most diverse groups of mammals in South America. So how long have neotropical primates been diversifying? Until recently, the earliest evidence for primates in America, at least for neotropical primates in America, were fossils from 26 million years ago in Bolivia, and these are called Gracile. But more recently, in 2016, Paropithecus was described at 36 million years ago. This matches well with the phylogenetic analysis. When you compare Catarrhini, the old world monkeys and apes, to the Platyrrhini, the split based on genetic data is at about 40 million years ago. So the fossils at 36 million years ago are pretty close to that rafting event. However, when we look at the standing diversity for extant neotropical primates, that divergence time is just at 23 million years ago, according to this time tree by Springer, suggesting that there were at least 15 million years of evolution of many different radiations of primates in that interim from the colonization of South America, most of all of which went extinct prior to present day, with only this one lineage with a common ancestor at 23 million years remaining. What's really interesting about this radiation in South America is that early primates arriving here were already pre-adapted to a forest niche that was not yet filled in the neotropics. So forward-facing eyes, ability to catch insect prey, grasping hands, ability to climb on branches to feed on fruit. These animals were able to immediately take advantage of forest habitats in a new way. And when we look at standing New World monkey or neotropical primate diversity, we divide this diversity into 5 families, and you can see that here with one representative depicted for each family. And so there are the Atellidae, which include the Morochis, howler monkeys, woolly monkeys, and spider monkeys, the Cebidae, which I'll be focusing on today, as we talked about with the squirrel monkeys and Capuchin monkeys. The Pithecidae include the Tedi monkeys, Sakis, bearded Sakis, and Wakaris. Aotidae include the night monkeys, and Callitrichidae include the marmosets and tamarins. And what's really interesting when we look at the fossil record at our best field site for the mid-Miocene in La Venta, Colombia, if we look at all the primate fossils that have been found from that site, and you can see the list of families here, all those 5 families are found at that site, and there are no primates at that site that can't be accommodated within these family groups. And so what that says is, and here I've constructed a phylogeny for neotropical primates by genus, and on the bottom you have a timeline. And so we're looking at their, on the far left, that first divergence is the crown extant Platyrrhini and the common ancestor to all present-day neotropical primates. And then we're going through time with splits leading up to the present diversity. And so if we mark on here a time slice, which is 13 to 12 million years ago when La Venta fossils were discovered, what you can see is that not only all the families had separated by then, but also there had been significant diversification at the genus level as well. And so it makes sense with the fossil record and the genetic information that by the mid-Miocene, we already had a pretty full complement of the primate diversity that we see today. And we can also map onto that phylogeny some of the key characteristics, key features of specific groups or genera, and see when along the timeline that they must have evolved. So we can see that prehensile tails evolved independently in the howler monkey group, as well as in capuchins. We can see seed predation and other feeding specializations have happened in different groups, and that tool use, for example, is a relatively recent innovation, although it's mostly been described by robust capuchins, but there are new observations of gracile capuchins using tools in the wild, so maybe that tool use is pushed back farther to the common ancestor of both types of capuchin monkey. So now we're going to think more specifically about phylogenetics and comparative biogeography. To understand primate evolution, we need to understand how taxa are related to each other and how long ago in time they diverged from each other. So just to give a few definitions here, I use genetic sampling of individuals to employ phylogenetic and phylogenomic techniques in order to understand how primates are related to one another, and then employ biogeographic methods to model how species have moved and spread across geography and biomes over time. And this field of neotropical primate phylogenetics and biogeography, with more complete studies across all genera and also with biogeographic modeling, has mostly occurred within the last 10 years. This includes both pan-platyrrhine studies, like these here, as well as more and more studies focused at the genus or family levels. These studies really got started with Collins and Dubach's work on spider monkeys and Cortez Ortiz's work with howler monkeys, and it has really expanded significantly and rapidly to include studies on almost all neotropical primate genera over the last 10 years. It's incredible, really, just being in this field and seeing how much we understand now compared to 10 years ago. And I wanted to give a shout out to many key researchers involved in this work. This is a meeting in 2012 at the International Primatological Society that led to a special issue on neotropical primate biogeography in MPE in 2015, including our review article on this topic. And some big questions that we discuss in this review relate importantly to the Cebidae. So I'm going to just give you some examples of questions of interest that kind of are across the neotropics, and then highlight a few aspects of the Cebidae before we really go into squirrel monkey and capuchin monkey biogeography. So one interesting thing that we found in the neotropics, a pattern, was that there are often sister genera with distinct distributions from one another in primates. One genus will be found just in the Amazon and the other genus in the Atlantic forest, which, as you can see here in the modern time, they're completely separated by these other habitats like the Gran Chaco, Cerrado, and Caatinga. So in general, these are dry forest, more open habitats. And so what we found by using genetics and biogeographic modeling is that these sister taxa all diversified from one another at between 5 to 10 million years ago, and that vicariance was likely a result of the growth of these drier habitat types. In fact, there are no new primate genera that enter the Atlantic forest after 4 million years. And so with all of these other taxa, the woolly monkey and moriqui, the Amazonian versus Atlantic TT, and the Amazonian versus Atlantic marmoset, you do see this distinction where one set is in the Amazon and the other is in the Atlantic forest, although the marmosets have also expanded out into the dry forest habitats. But with the capuchin monkeys, the gracile capuchin only comes down to the border of the Amazon. But in fact, the robust capuchin, while it's found throughout the Atlantic forest. It's also found widely spread in the dry forest habitats as well as the Amazon. So we'll talk more about what happened there in a little bit. But in general, we found this pattern that primates have been diversifying in the Amazon since 20 million years ago, that they expanded into the Atlantic forest in the Miocene, with subsequent variants with the drying out of those forests into more savanna-like habitats. And then no new primate genera arrived in the Atlantic forest after about 4 million years ago. So it was effectively kind of blocked off from the Amazon. Another key moment in neotropical primate evolutionary history was 3 million years ago, when South America stopped being an island continent and came in contact with the formation of the Isthmus of Panama, came into contact with Central Meso North America. And what you saw at that time, at like kind of the simple story, is that at 3 million years ago, everyone lined up and just funneled through north and south. So you have all these species from the south invading north and vice versa. But what we were able to do by looking at the dating of the splits between Southern and Central American taxa of primates was just to see if it really was the same timing for all primate groups. And what we found was that, in fact, the spider monkeys and howler monkeys do look like they were part of that Gaby interchange right at 3 million years ago, but that the Capuchin monkeys and the squirrel monkeys were both later arrivals, with Capuchin monkeys looking to have colonized Central America about 2 million years ago, and squirrel monkeys even later at about 1 million years ago. And it's interesting also to think about how primates really were affected by this interchange. One thing I found interesting in terms of the behavioral ecology is that despite the influx of North American competitors and predators, monkeys still have the greatest predation rates from raptors, which would have already been in place for a long time, and their behavioral strategies still seem to be for raptors, like their primary strategies for predation defense. What's also interesting is despite all those different types of animals that came down from the north, none of these really fits well into a primate niche. Maybe the squirrel would be the closest, and some other rodent species. But really nothing outcompetes primates for that gregarious diurnal arboreal niche. And so primates and birds continue to dominate the diurnal neotropical landscape in forest. And even though there were these waves of primate colonization up into Central America as far as Mexico, no primates were successful in moving past those desert environments to invade North America. And then the third thing that I wanted to talk a little bit about was those dry forests that I mentioned. So usually when we think about primates in the neotropics, we think about rainforests. We think about the Amazon basin, the Atlantic forest, the forests up in Central America. But it turns out that primates, and this map, the colored section of this map, is the areas that primates inhabit in the neotropics, and the white part without color is not part of their current distribution, although just to mention that those yellow dots are all fossil localities. So anyway, we often think about the rainforest, but what about these dry forests? Another question that we had was, when did primates colonize these dry forests? And is there continuous occupation of these drier areas, or is something else happening? And so from our research, what we found, with one exception, which is a howler monkey that seems to have colonized and occupied dry forest habitat over the last 4 million years, that all of the other extant primate species expanded into savanna habitats recently, less than 1 million years ago. And so our interpretation is that the pattern appears to be continuous occupation of rainforest with these repeated waves of expansion and extirpation in savanna habitats. And so here you can see the timing of splits for these species that have moved into drier areas. And again, just to focus on the Cebidae, you can see Cymeri albigena, which occupies the Colombian llanos and is really the only squirrel monkey species, as far as we know, that uses a significant amount of dry forest. And then also Cebus olivaceus is found in the Llanos and Roraima savanna areas. And here this depicts Sapajus xanthosternus, which uses both Atlantic forest but goes into dry forest habitat as well in the Cerrado. And interestingly, the species that has the widest distribution in the dry forest is Sapajus libidinosus, depicted here using stone tools. And stone tools is habitual and common across many populations of libidinosus in these dry Caatinga and Cerrado habitats. And as we'll see when I talk a little more about the biogeography, they're a pretty recent invasion, just like I was saying, into the dry habitat, and yet when we look at their morphological characteristic, there seem to be adaptations such as shorter tail and different limb proportions that are specialized for this dry environment, using the ground more often. Okay, so now let's take a look specifically at squirrel monkeys. And I'll talk about my research with collaborators, and here it gives a nice overview of the diversity of squirrel monkeys. As Humboldt himself described, squirrel monkeys are extremely charismatic, and as he said, they're often favorites of local people due to their beauty, their small size, and their gentle behaviors. One thing I want you to notice as you look across the diversity of squirrel monkeys depicted here, across the Amazon and into Central America, is that, and I just want to shout out to Amisha Gadani, who made these drawings. She is my collaborator at UCLA. But I want you to notice that they're all very similar in shape, size, and even coat pattern. It's subtle variations that distinguish across these described species. And here's the distribution, so you can see mostly an Amazonian distribution, but if you look up into Central America, there's a very small restricted distribution in Costa Rica and Panama on the coast, which is Cymeri orstedi. And in fact, so here you can see many different species, which I'll continue using this diversity of species, but you should know that squirrel monkey taxonomy has been highly contested based on morphology, ranging from about 1 to 8 different species recognized. But for now, I'm going to go with Philip Hershkovitz species hypotheses. And I want to just highlight him as an extremely important researcher, explorer, and taxonomist working with the Cebidae and more broadly with neotropical primates. And so Hershkovitz hypotheses based on morphology separated squirrel monkeys primarily based on their Roman straight across arch versus their column-shaped Gothic arch on the forehead coat pattern. And I'll show you that Boliviensis had a Roman arch, and Sirius had the Gothic arch. Hershkovitz also separated out a species, the Costa Rican and Panamanian species orstedi, and Ustis he separated, which are very distinctive with bare ears, whereas the rest of these species have tufted ears. So here you can see Boliviensis with the Roman arch, Orstedi with the Gothic arch, Ustus with the bare ear, Sirius with the tufted ear. And so to go into some of the genetic work, one of our first studies was looking at mitochondrial genomes across a few of these species and subspecies. And we did find this basic pattern of Gothic arch versus Roman arch as the major split, at least with the taxa that we had to work with. But we also saw some discrepancy with the Hershkovitz hypothesis in that two subspecies of Sirius didn't form a clade together, but instead Sirius from the Guianas was more closely related to Orstedi from Central America than to Macrodon from the Western Amazon. What was also pretty surprising, although it kind of makes sense when you think about that very conservative squirrel monkey morphology, was that crown cymeri, the divergence time for all extant squirrel monkey taxa, using two methods, multi-div time and beast, was found to be just somewhere between 1 and 1.5 million years ago. And so that's pretty recent, and in fact, some phylogeneticists have argued that species should only be considered real species if their divergence time is at 2 million years or earlier, which would in fact put cymeri all together, lumped as one thing. So we wanted to explore this in more detail, and we did work with mitochondrial DNA, but a much larger sampling of individuals and species. This study, which also used mitochondrial DNA, was an international collaboration with lab work done by Gohar Kashkarian and Mai Wen at UCLA and Michel Mercedes in Brazil, and fieldwork done by multiple people, including Fernanda Paim, who headed the project for the part at Mamirawa. And we also were using museum specimens for a lot of the samples as well. So just to mention Mamirawa in a little more detail, the site Mamirawa is by Tefe in Brazil, and it was especially important because it was the only locality for a relatively newly described species of squirrel monkey, Cymeri vanzolini. Morphologically, vanzolini has the Roman arch, like Boliviensis, but it is also syntopic in its distribution with two other squirrel monkey species, Macrodon and Cassiquariensis. So it's right at the crossroads for three other species distributions, if you look at the map. And so this is the major biogeographic analysis that we did in the study. And in this biogeographic reconstruction of squirrel monkey evolution, you can see on the left the color coding for the different regions which are used within the tree. So this is a time tree, and the colors signify the locations of those ancestral populations through time. And so what you can see is that many clades are of a given color, and so that's showing the geographic signature of the data. But on the other hand, what you'll see is that the morphological patterns don't actually match very well with the relationships we found in the mitochondrial DNA topology. So to give you some examples, Boliviensis did not come out as a sister group to Vanzolini, even though they both have Roman arches, and I'm highlighting along the right side the species names for the different clades. So there, Boliviensis and Vanzolini came out separately in the phylogeny. Ustus with the bare ear didn't come out as one single group, but instead came out in multiple places on the phylogeny. And then Sirius, which is the far eastern Amazon, the Guiana region, was considered prior to some of this work to go not only in the Guiana region, but also to come down into the Xingu and Belém regions in southern Amazon. But here we're calling that type Collinsy, and Collinsy and Sirius also did not come out in the same place together as a clade in the phylogeny. So in fact, we got really different results than what we were expecting, and it did not match Hershkovitz's morphological hypotheses. However, when we took the same data, as I showed you already, you can see this geographic signature. When we constructed minimum convex polygons based on all the samples that had clade membership, we did see this very strong geographical structure, and you can see, for example, strong evidence of the Amazon River as a barrier to squirrel monkey gene flow. With these confusing mitochondrial DNA results, we decided to continue research with genomic sampling, nuclear markers using dd-RADS, which is a RAD-seq technique. And so Michelle Mercedes, who you can see in the top photograph, and she's on the bottom left of that photograph, she had worked on the mitochondrial DNA project, and now she has written her doctoral dissertation on this nuclear genomic project. And I want to also shout out to Hazel Byrne, who headed up the lab and worked in very close collaboration with Michelle on this project. And this map is just depicting the locations of samples that were used for the dd-RADS study. And you can see in the inset, that's that Tefe region that has multiple different species present. From this data set with nuclear DNA, we got a topology that mapped perfectly with the morphology hypothesis of Hershkovitz. So you can see the first split is between Roman and Gothic. The second split is between bare-eared and tufted-eared. And then Cassiquariensis comes off in the western Amazon, next Macrodon, also in the western Amazon, and then you have the East Amazonian clade. So here we see Collinsii and Squirrel grouped together, unlike what we saw in the mitochondrial DNA. Here we also see Boliviensis and Vanzolini together, unlike the mitochondrial DNA, and we see all the Oostis together. And so I think it's really important to consider both the mitochondrial and nuclear data and understand how they complement each other and what they can tell us together. For this data set, the nuclear data set, we also performed a structure analysis, which shows the main clusters within the data set. And here we also see the same pattern of the Roman versus Gothic, the bare-eared versus tufted-eared, the Northwest Amazon, Southwest Amazon, and Eastern Amazon as the major different groups within squirrel monkeys. Something else to notice, however, is the introgression or evidence for hybridization that you can see in the structure analysis. So each line in the figure represents one individual, and you can see that for some individuals, their color is not just one color, but they have multiple colors in their line, and that's showing the proportion of that individual's genome that comes from the different colored populations. So if you just focus on Cassiquariensis and Macrodon 2, what you can see is that Cassiquariensis individuals are mostly red, but they have some purple. And Macrodon 2, some individuals are all purple, but some have some red. And that purple and red mix suggests that there has been hybridization or gene flow between Cassiquariensis and this group called Macrodon 2. So to summarize the findings from both the mitochondrial and nuclear DNA and what that kind of says about Cymeri biogeography, we can say that Cymeri has diversified very recently across the Amazon and into Central America within the last 15 million years. And what's very cool about that is that if you look at the fossil record, we have Neo-Cymeri, which some now call Cymeri because it's so similar to modern-day Cymeri, but coming from La Venta, which I talked about, at 12.5 million years ago. So there's a very conservative morphology and yet the most recent radiation of these squirrel monkeys has happened just in the last 1.5 million years. So it suggests this repeated replacement of taxa despite retaining a similar morphology. And what we saw was that the nuclear markers match really well the Hershkovitz morphological assessment of Roman versus Gothic arch squirrel monkeys and bear versus tufted-eared dichotomies. But what the mitochondrial DNA markers add, even though they seemed confusing at first, was they add evidence that there's been rapid and incomplete diversification of squirrel monkeys. So we have this nuclear pattern of how the original diversifications happened over time and across rivers. And then what we see as squirrel monkey species expand their range, they come into contact with some of those other species, and you have hybridization events. So I think that's really interesting, especially there's this pattern of hybridization across much of the southern Amazon. Okay, now I'm going to focus on the Capuchin monkeys, and we'll start looking at the group as a whole, and then we'll take a closer look at each group by itself. And so all Capuchins were lumped together in one genus, Cebus, for about 50 years, and only recently they've been split into two genera. And that work, that really important work based on morphology, was by Silva Jr. in Brazil, and his nickname, or he's better known as Cazuza. So in this map, what you can see is the distribution of Capuchin monkeys, and in blue you just have Gracile Capuchin monkeys, in yellow just robust, but in all the green area, which is most of the Amazon, you have both Gracile and robust Capuchin monkeys in symmetry. And so we started this project of looking at Capuchin monkeys in order to try to understand the symmetry between robust and Gracile Capuchins. Our time tree revealed a divergence time between Gracile and robust Capuchins of about 6 million years, about the same amount of time as the split between chimpanzees and humans. In the biogeographic analysis, we see diversification over time, moving from the initial divergence between Cebus and Sapajous on the far left, closer to present going right, and the colors on the branches indicate the most likely region that these ancestral populations were found in. Cebus has been diversifying within both the Andes and the Amazon for 2.5 million years and expanded to Central America as early as 2 million years. You can see these blues and greens are that part of the Neotropics. And then in contrast, for most of Sapajous' evolutionary history, they were diversifying within the Atlantic forest, the red and orange colors, and only very recently, within the last 500 years, in this analysis, have they expanded into the Amazon basin and the Cerrado habitat. This was an extremely rapid Pleistocene range expansion. So our new understanding of Capuchin monkey dynamics is that they, like several other neotropical primate genera, split between Amazon and Atlantic forest populations through vicariance due to drying out of the central region. But then much later, Sapajous is able to expand back into both the drier forests and between the two rainforest habitats, and to recolonize the Amazon into widespread symmetry with Cebus. We followed up on this with two subsequent articles, one that expanded our work with mitochondrial DNA and a second which utilized ultra-conserved elements, a nuclear genomic sampling technique. These papers were first authored by Marcela Lima on the left as part of her doctoral dissertation, and she worked in strong collaboration with Janet Buckner on the right at UCLA when she came to my lab as part of a Science Without Borders sandwich grant. Here's a depiction of our sampling for the second mitochondrial DNA paper, as well as the biogeographic regions we characterized in our biogeographic analysis. And so here, using Biogeobear's biogeographic analysis, we confirmed this pattern with more samples where Cebus diversified first in the Amazon quickly expanding to the Andes and Central America, whereas Sapajous diversified first in the Amazon and later expanded into the dry forest Here, using a biogeographic analysis, we confirmed the pattern with more samples, showing again that Cebus diversified first in the Amazon and quickly expanded to the Andes and Central American regions, while Sapajus diversified first in the Atlantic forest and much later spread through the Amazon and dry forest regions. A phylogeographic analysis in beast phylogeography for the cosmopolitan group of Sapajus is shown here, with more fine-grained regional separation for the Amazon region. It shows the sister species to the Amazon cosmopolitan cluster is Sapajus robustus, and that the initial expansion into the Amazon was in the Rondonia region, but that there have been multiple subsequent expansions into the Guiana, Negro, and Inambari regions, as well as into the Cerrado Caatinga and back to the Atlantic forest. This phylogeographic analysis suggests that Amazon rivers are weak barriers to Sapajus gene flow, and that Sapajus has the capacity to adapt to diverse habitat types. Now moving to the nuclear genomic study using UCEs, we also found that a SNP phylogeny grouped Sapajus in the Amazon in geographic clades, but that these did not map on well to hypothesized morphospecies. So a northern clade included samples from Macrocephalus and Apella, and a western clade also included samples from Apella, Macrocephalus, and Kai, whereas an eastern Amazon Caatinga Cerrado clade included Apella again and Libidinosus, with Flavius as the sister group to that clade. Many of these cosmopolitan clades also have relatively weak support values, probably a reflection of their relative rapid and widespread diversification with little barriers to gene flow. I'm going to show you a couple of structure analyses for population clusters for Sapajus. The first one we did not permit admixture and reveals two main clusters, one Atlantic forest and the other Amazon and dry forest. Within these main clusters separates into two more clusters each that fits well by biome type, separating Amazon from dry forest and North and South Atlantic forest. However, once you allow for admixture in the structure analysis, we get four clusters, but there is widespread evidence. So if you see a whole circle with one color, there's no admixture. If there's a pie with a pie piece of a different color, or two pie pieces additional, those are all signs of admixture. Okay. And we saw population admixture in all regions except for the northern Atlantic forest. Both the UCE and MTA DNA data are concordant in showing regional subgrouping within the Amazon with northern and western clades, but both also provide evidence for substantial introgression or hybridization, and neither show strong association with morphological species concepts, but instead suggest that there's relatively little differentiation. Here comparing results from both papers. Both the UCE and mtDNA are concordant in showing regional subgrouping within the Amazon with northern and western clades. But both also provide evidence for substantial introgression or hybridization, and neither show strong association with morphological species, but instead suggest that there is relatively little differentiation across Apella, Macrocephalus, Kai, Libidinosus, and Flavius. As I mentioned, for about 50 years, Cebus apella was considered to be the one single robust or tufted Capuchin species, and was spread throughout the South American range. Subsequently, morphological work by Silva Jr. and Groves pointed especially to increased diversity in phenotypes within the Atlantic forest, and the number of species grew to 8. In Lima et al. 2018, we suggest reducing the number of species of Sapajus. At our most extreme suggestion, we say to reduce it to 4. And in this scenario, Sapajus apella would return to having by far the largest distribution for any neotropical primate species. Okay, now we're going to switch over to the Gracile capuchins. While sapajous expanded in number of species, Cebus has remained relatively stable. Hershkovitz in 1949 recognized 3 gracile capuchin species, Capucinus, olivaceus, and albifrons, and then Kaipori was discovered in 1992 in far eastern Amazonia. It has both been considered its own species and sometimes a subspecies of olivaceus. In collaboration with 2 other giants in biogeography and taxonomy, Jean Boubly, it's Anthony Rylands on the left and Jean Boubly on the right, we performed a preliminary analysis of Cebus genetic diversity, here again using mitochondrial DNA. In these analyses, we found geographic clades for taxonomic groups, but they didn't map well to the species hypotheses of Hershkovitz because some albifron subspecies were more closely related to olivaceus and others were more closely related to Kaipori. So just to go back to Hershkovitz, he was an extreme lumper and he described several new forms of albifrons, but at the same time lumped them all together as subspecies with other subspecies he already knew about for 13 subspecies of albifrons. And here, just looking at the albifrons distribution, you can see it may be easier if I take away the other species. And what you can see is this disjunct distribution of 4 separate areas that correspond to the Amazonian basin, the Colombian and Venezuelan Andes, and then the western coast of Ecuador and going into Peru on the other side of the Andes, and up at top you can see the small population on the island of Trinidad. And if we locate the holotypes for the different subspecies, we can see a high concentration of these species in the Andes compared to the much larger distribution in the Amazon with fewer subspecies. And so in the paper headed by Marcelo Lima of mitochondrial DNA in 2017, looking at Cebus variation, what we saw, and I'm just going to map it back onto this same 4 parts of the albifrons distribution, but I'm including the Capucinus and olivaceus and Kaipori samples as well. We can see we came out with these major clades within Cebus. So olivaceus and Kaipori came out as one clade in the Guiana region going into eastern Amazon. There were 2 other western Amazon clades. There were 2 Andes clades, and there were 2 Central American clades. And then these clades in the Andes and Central America actually grouped together as one large clade. But this mitochondrial DNA study didn't clearly delineate the hierarchy of relationships across these 4 clades because they came out as a polytomy in the tree. Lima's 2018 paper included Cebus species as well, and here at the top you can see the UCE species tree that puts Kaipori and olivaceus as sister species and Capucinus and albifron/unicolor as sister species. But we really need many more samples from many more locations in order to really understand the dynamics of Cebus evolution with nuclear data. So I think what the genetic studies did, at least in terms of my own experience, was really pique my interest to know more about the morphology of Cebus and to look at the geography in more detail. So in 2016, we formed an international collaboration at the International Primatological Society meetings in Chicago with the hope to gather information and photos of Cebus from known locations and to review all the literature to try to understand what was going on with the biodiversity of this group. And so we now have a very large collaboration group. This isn't even all the people. They span the geographic range of Cebus. And I want to shout out particularly to some Colombian collaborators, including Diana Guzman, Kimberly Negrete, and Andres Link, pictured here from left to right with me in there too. And also mention Ivan Dario Soto Calderon, Pablo Stevenson, and Sebastian Garcia. So I'm just going to take you through some photos so you can get a sense for Cebus diversity based on this collaboration and photos that we've gathered together, and then I'll show you a few other things. So these photos are all a series of 3 with a juvenile, an adult male and adult female. First is imitator from Central America, Malatiosis from most northern Colombia, Cesari, this beautiful, highly diverse, light-colored fur. Each individual is variable. Trinitatis from the island of Trinidad. Versicolor, which has been studied on an ongoing long-term field site by Andres Link and several students, including Kimberly and Sebastian. Leucocephalus, these photos are from Venezuela on the Lake Maracaibo by Alan Heighton. Equatorialis in western Ecuador and Urachus in eastern Ecuador. Albifrons and unicolor, which I'm following Deffler, another giant in primate taxonomy in the Neotropics, who Deffler and Camacho have this paper describing the neotype for albifrons and suggesting that albifrons and unicolors should be lumped together. And based on my research so far, I agree with that idea. Here's Tom being celebrated for his contributions at the Colombian mammalogy meetings in 2018. A few more species, Cucinus in Peru, Olivaceous, these are Venezuelan Olivaceous. On top here are Olivaceous from Brazil and Suriname, and then Kaipori in far eastern Amazon and Brazil. And so we're using this information from skins, from the literature, from live photos to try to understand the diversity across distributions, putting this information together to hopefully to have kind of a field guide article that looks across biodiversity in Cebus. And I'm collaborating again with Amisha Gadani for these drawings. Here you can see the holotype locations and the morphology across the Neotropics for Cebus. There's another view of this diversity kind of schematically laid out in a geographic sense. And I think the thing that's interesting, for example, if we look at Colombia, on the one hand, we have this story of sapajous coming from the Atlantic forest and expanding range out, so its far edges are into Colombia, and there's only one species in Colombia, which is this pan-Amazon species Apella, and on the other hand, you have this extreme diversity of forms and types of Cebus in Colombia. So Colombia has the most species, at least in that Hershkovitz sense of Cebus. And I think thinking about Capuchin monkeys who are able to cross rivers and seem to have gene flow across wide ranges in the Amazon, but it's really interesting to try to think about the geographic barriers of mountain structures and how that has led to diversification for Cebus in the Andes. Okay, the last thing that I wanted to just talk about for a minute was the state of Cebid biodiversity and how the phylogenetic and biogeographic studies change taxonomy, and that itself has conservation implications. So it turns out that many of the same people that work on phylogenetics and biogeography are also the same people that are in the primate specialist group and work for IUCN to do the red listing to decide which species are endangered and at what level. This is us in Texas at the IUCN Neotropical primate red listing. And so one species of special concern is Cymeri vanzolini. With our molecular studies, we show that it is a unique taxon. And what we've also done in terms of red listing is to list it as endangered. And this is one of the first species I know of that it's listed as endangered because its entire habitat is subject to, in climate change modeling, will be subject to more and more extreme flooding events. So the entire habitat where it lives will have a change in cycle that may lead to it not being able to survive there. Our studies also place Sapajus xanthosternus as the kind of most unique Sapajus taxon, the one that's split off first, and it is listed as critically endangered. But if we think about our biogeographic studies and this recommendation to lump back all these species into Apella, then what does that mean for conservation of a species like Sapajus flavius? Flavius has a very restricted distribution in Atlantic forest and Caatinga, few populations left, and it is morphologically distinct, even with a barbella, a throat sac that's not seen in any other species. So even if it is genetically similar and recently evolved, what does that mean for conservation? And right now, Amelie Martins in Brazil is doing her PhD on trying to understand conservation genetics for this species. But I think like the biggest change really is for Cebus species. Caipori in Brazil is critically endangered. It's in the arc of deforestation, and it's so rare in its habitat they don't know the boundaries of the habitat because it's just incredibly hard to find. And then the most changes really will be in understanding better these subspecies for Hershkovitz for Cebus, and the process has already begun to happen with the last IUCN meeting of separating several of these subspecies into their own species, but I think the work hasn't been finished. So we're like on a precautionary measure. We are splitting, maybe over-splitting, and part of the work that I want to keep doing with these morphological and geographic analyses and more genetic studies is to really see, are all the species that are described from the Magdalena Valley really one thing, or are those actually 4 species that you find there? Just as an example, is albifrons and unicolor the same thing, or are there strong genetic signatures? But for certain, the Andes species and Equatorialis and Trinitatis need urgent attention. Okay, thank you very much. I'd like to thank you one more time for coming to my talk today, and to thank the International Biogeography Society for putting on this event for International Humboldt Day 2020.